in each and every department. Today, our Department of Plant Pathology and Entomology is organizing a webinar on conservation, biological control, and biopesticides in agriculture. We contacted so many scientists, and 5 scientists and 5 eminent scientists accepted our invitation. And I am thankful to all our keynote speakers who accepted our invitation. Today, we have Dr. Pramod Mal as chief guest and a keynote speaker from Panagar University, Dr. Shankar Ganesh, Central Institute of Cotton Research, Coimbatore, Dr. Muhammad Akram, Principal Scientist at IIPR, Kanpur, Dr. P.K. Mishra, he's also from IIPR, Dr. Kamal Khiladi, who is professor at Sardar Vallabhbhai Patel University of Agriculture and Technology, Modipura. As we all know, modern agriculture systems during the last 50, 60 years have largely followed substitution agriculture, in which weed, pest, and disease control is usually achieved by agrichemical use. That has caused severe environmental harm, including reductions in biodiversity. This type of agriculture is increasingly becoming unsustainable. Pressure for sustainable practices, both through markets and regulatory bodies, and the increasing problem of pesticides resistance are industry to find alternatives to agri-chemicals. Currently, such alternatives include plant-derived essential oils, plant defense stimulants, biopesticides, and conservation biological control techniques. At this time, conservation, biological control, and biopesticide are being used in narrow niches of agriculture and are generally effective with an understanding of the system and its limitations. Research is now underway to make both these techniques more effective under a wider range of environmental conditions, pest, species, and cropping systems. Among the major benefits of incorporating biopesticides into a sustainable agriculture system is that they are generally more environmentally friendly and do not damage the soil, water supply, or the wildlife. Including the beneficial insects, the importance of nurturing a standing army of beneficial insects can't be overstated. So our keynote speakers who are today, who are very eminent in their field, will deliver their lecture on relevant topics. As we all know that today's webinar is organizing only because of our principal who always guide us and who always motivate us. I would like to invite Professor Naresh Kumar, Principal of Chaudhry Choturam Postgraduate College, to inaugurate and to welcome all the participants. Thank you, sir. Principal, sir.
Thank you, Dr. Arun. आवाज आ रही है ना? हेलो, आवाज आ रही है?
जी सर, आ रही है.
अच्छा. Thank you, Dr. Arun. Chief guest of this national webinar, Professor Pramod Mal, former head Department of Agriculture and Entomology, Govind Ballabh Pant, University of Agriculture and Technology, Panagar. Our eminent speakers, Dr. Muhammad Akram, Principal Scientist, Indian Institute of Pulses Research, Kanpur. Dr. R.K. Mishra, again Principal Scientist, IIPR, Indian Institute of Pulses Research, Kanpur. Professor Kamal Khiladi, Department of Plant Pathology, Sardar Vallabhbhai Patel University of Agriculture and Technology, Modipura, Meerut. And Dr. K. Shankar Ganesh, Senior Scientist, Central Institute of Cotton Research, Coimbatore, Tamil Nadu. Convener of this webinar, our Head of the Department of Plant Pathology, Dr. Arun Kumar ji.
17 number.
Organizing Secretary, Dr. Joni Kumar, Assistant Professor, Agriculture and Entomology. Co-organizing Secretary, Dr. Dushyant Kumar, Assistant Professor, plant pathology, distinguished participants, guests, friends, dear students, ladies and gentlemen. First of all, I congratulate our chief guest of today's webinar and all eminent speakers as well as participants on behalf of our college from each and every nook and corner of my heart. It is a matter of great privilege for us that today we have a galaxy of eminent speakers in this webinar. And these speakers, these are from prestigious institutes of our country. Now the credit for this that goes to Dr. Arun Kumar, convener of this webinar, Dr. Joni Kumar and Dr. Dushant Kumar, two of our colleagues. These are very new incumbents, Dr. Joni Kumar and Dr. Dushant Kumar, and they in collaboration with Dr. Arun Kumar have taken pains to organize this national webinar on very important topic. I congratulate all of my friends, my colleagues for organizing this webinar. The topic, as Dr. Arun Kumar has said, the topic of this webinar that is very relevant, very important, that is conservation, biological control and biopesticides in agriculture. I will not say much about this topic because I am also a student of botany. So some in applications of biology we also read about this topic. और मैं अपने विद्यार्थियों के लिए खास तौर पे जो इसमें जुड़े हुए हैं उनके लिए मैं खास तौर पे एक दो बात उसमें कहना चाहता हूं कि एक होता है टर्म पेस्ट, आप जानते हैं पढ़ते हैं प्लांट पैथोलॉजी एंटोमोलॉजी में, प्लांट प्रोटेक्शन में, that is epidemic disease causing organisms. They may be bacteria, they may be insects, they may be fungi, they may be weeds, all of these are pests. ये सब pests. And the chemicals used for a long time for killing these pests, that is pesticides. Or these are also known as biocides. Sides कहते हैं मारने वाले को, bio यानी कि पेस्ट लिविंग ऑर्गेनिजम्स को मारने वाले को जो नुकसान पहुंचाते हैं फसलों को हमारी. These are what pests. Now these pesticides, for example, Bordeaux mixture, malathion, DDT, furadon, these are very different groups, organochlorines, organocarbons, carbamates, pyrethroids, many types of these pesticides are there. But the main difficulty with these pesticides is that they cause health hazard. They are biological because they are non-biodegradable and therefore they are biologically magnified. That is, their concentration goes on increasing with each and every trophic level. So biomagnification occurs, and that is a hazard, environmental pollution. That is environmental pollution. So the alternative, as Dr. Arun Kumar has said, of this, that is biological control. That is use of other living organisms to kill these pests. That is biological control. And biopesticides. Biopesticides, they may be bioherbicides, they may be bioweedicides, rodenticides. This is the common term, that is biopesticides. Now, this is very important topic, that is conservation, biological control. That is how the principles of ecology and conservation biology, these are used to maximize biological control. This comes under conservation, biological control, and it presents various methods to modify or to manipulate the environment so as to accelerate or so as to enhance the activity of the natural animals of the pest. यानी कि एनवायरमेंट को ऐसा बनाना, इस तरीके से मॉडिफाई करना. हमारे बच्चे खास तौर पे इस बात को समझे, बाकी तो एमिनेंट साइंटिस्ट हैं, हमारे स्पीकर्स हैं, वो आपको डिटेल में बताएंगे. यानी कि ये जो टॉपिक है ये बताता है कि कैसे एनवायरनमेंट को मॉडिफाई किया जाए, कैसे मैनिपुलेट किया जाए ताकि नेचुरल एनिमीज जो पेस्ट के हैं उनकी एक्टिविटीज को एनहांस कर दिया जाए, ताकि वो जो है उन पेस्ट को खत्म कर दे. सो दिस इज द टॉपिक टुडे which has been taken by our learned friend Dr. Arun Kumar, and I congratulate him especially, although young colleagues are also associated with him. But this is a very important topic, and we ourselves as well as our students, all the participants, they will be benefited to a great extent. I am hopeful that this webinar will be achieving its goal. Whatever is our objective of this webinar, that will be achieved today. This is my thought. This will happen. And not taking much time again, I once again congratulate. मैं अभिनंदन करता हूँ, congratulate करता हूँ, बधाई देता हूँ और अपने गेस्ट का अभिनंदन करता हूँ पूरे महाविद्यालय परिवार की तरफ से, जिन्होंने अपने बहुमूल्य समय में से ये समय निकाला, ताकि, क्योंकि मेरी सोच ये है कि बड़े-बड़े अच्छे-अच्छे इंस्टीट्यूट्स, चाहे हमारे देश के हैं चाहे बाहर के हैं, उनके अच्छे-अच्छे साइंटिस्ट हमारे बीच में आए. और इसी कड़ी में ये आज वेबिनार प्लांट प्रोटेक्शन का यहाँ पे किया जा रहा है. तो मैं अपने गेस्ट का, अपने एमिनेंट स्पीकर्स का यहाँ पे पुनः बहुत बहुत धन्यवाद करता हूँ, अभिनंदन करता हूँ कि हमारे लिए उन्होंने समय निकाला. थैंक यू वेरी मच. जय हिंद, जय भारत.
Thank you very much, sir. Thanks a lot. We always have grateful. आप इतने अच्छे शब्दों से हम लोगों को आशीर्वाद देते हैं और प्रोत्साहन देते हैं. मैं अपने सभी पार्टिसिपेंट्स को बताना चाहता हूं कि हमारा एग्रीकल्चर कॉलेज एक भारत में एक बहुत अच्छा स्थान रखता है और हमारे प्रिंसिपल साहब लगातार मोटिवेट करते हैं कि हम लोग उसमें बढ़ चढ़ के काम करें और स्टूडेंट्स को जितना ज्यादा से ज्यादा हम लोग दे सकें, उतना हमारे प्रिंसिपल साहब हमको मोटिवेट करते हैं. आई एम हाईली थैंकफुल टू यू सर, and you just gave a beautiful words to all of us. Thank you very much, sir. Thank you very much. In this webinar series, we have our chief guest, Dr. Pramod Mal sir here. Sudhir ji, Dr. Pramod Mal sir हमारे साथ हैं?
Yes, sir. He is available.
Dr. Professor Pramod Mal sir, professor at Department of Entomology, Govind Ballabh Pant University of Agriculture and Technology, Pantnagar. He is former head of the Department of Entomology. I would like to invite Dr. Mal sir to say a few words as chief guest at this webinar, sir. Dr. Mal sir.
Thank you, Arun Kumar ji, Dr. Arun Kumar ji. Very good morning to all of you, all the dignitaries sharing virtual dais on this great occasion. Great and auspicious occasion. Our esteemed fellow Dr. Muhammad Akram, who is principal scientist, IIPR, ICR Kanpur. Dr. R.K. Mishra, a principal scientist, IIPR, ICR Kanpur. Professor Kamal Khiladi Sahab, he was my senior earlier, Department of Plant Pathology. Sardar Vallabhbhai Patel, University of Agriculture and Technology, Modipuram, Meerut. Dr. K. Shankar Ganesh, Senior Scientist, ICR, Coimbatore, Tamil Nadu. He was my student earlier. Host dignitaries, the patron Professor Naresh Kumar ji, principal of this event, and enriched us a lot about this biocontrol or biological control, bioagents. Convener Dr. Arun Kumar ji, Professor Plant Pathology, and co-organizing secretary Dr. Dushyant Kumar, Assistant Professor Plant Pathology. Organizing secretary Dr. Johnny Kumar, Assistant Professor Entomology. First of all, I would like to thanks the patron, Professor Naresh Kumar ji, convener Dr. Arun Kumarji, and especially Dr. Jani Kumar, organizing secretary, for rendering this great opportunity to share the dais with you, venerable fellows. It's my great pleasure to be the chief guest of this great event, National Webinar on Conservation, Biological Control, and Pesticide in Agriculture. This event would indeed be worthwhile in order to conserve, promote, augment the bioagents for sustainable agriculture. The people participating in this national seminar will be acquainted with various agronomical practices which are performed, would be critical for pest management if endowed in consideration to save our bioagents. Of course, the eminent scientists are participating in this event, delivering useful lectures on bioagents and biopesticide would be the immense importance. I am thankful to all organizing team for remembering me and organizing such a valuable event to strengthen our ecosystem for sustainable agriculture. Friends, this is the matter of serious concern for all of us, those who are engaged in doing research or managing the pest population, evolving some methods which are useful to pest management. The use of the chemicals or so-called modern agriculture has created a lot of problem before us to save our planet, to save our ecosystem, or strengthen the biodiversity which are available with us. But this great occasion and the thinker who started this today's event will be of great importance, because the of course we have produced a lot of things. We have produced a food grain, but surplus food grain we are producing, and that is more than sufficient for our survival. 2 years or 3 years, if no grains are being produced in our country, we can survive on the surplus food grains which have already been produced. But the thing is that now the concern is that the quality of the food grain, because we are concerned about our impaired health, the loss which is made to the environment by using the chemicals or pesticide indiscriminately and excessive way, and which has killed the most of the component, are killing, you can say, if any component of the ecosystem is being deteriorated, or if we are damaging that component, ultimately we will have to pay. So this is not going to pave the way if we are using the chemical and suppressing or managing the pest population. This is the matter of concern, and now the entire focus has directed towards the minimum use of the chemicals, and how the minimum use of the chemical can be good for sustainable agriculture if we are managing all these the bioagents or microbes which are useful for management of the pest, for causing disease in the pest. So many biorational components are there in the IPM. Now these are the components which needs to be emphasized in the IPM. The various bioproducts are available, and of course some semiochemicals are there which are playing, of course, important role. We will have to explore the potential of these biorational compounds, and that can be utilized in eco-friendly way to suppress the pest population and save our health without causing any hazards to the environment or any hazards to our natural enemies or the friend insects or microbes. So with this, we will discuss in detail. I think this is the welcome. This is included in welcome address. So we will later discuss each and every component in details. So once again, I am thankful to all these dignitaries which have organized, the host patrons, especially Professor Naresh Kumarji, who has organized such an event in his supervision and direction. And Professor Arun Kumarji, you also deserve thanks for this great event. And my student Dr. John is there, so I am happy, and this is proud moment for me that he is doing such a valuable job or the activities in that Muzaffarnagar college. So thank you once again for just giving me this great opportunity.
Thank you, Professor Mal sir, for a great words. As we all know, we are just having a series of webinars here. Our principals says that you just do in a series, and it will be beneficial for all the students, our faculty members and others. So we decided to do this job here. And in this series, we have very eminent speakers here. Professor Dr. Muhammad Akram sir, who is a principal scientist at Indian Institute of Pulse Research, Kalyanpur, Kanpur. I would like to introduce him. He had 6 years teaching experience at CS University, Kanpur. He has attended almost 8 training programs and published more than 50 research papers in reputed journals. He has published more than 50 popular articles in different magazines and 10 book chapters. He has attended more than 35 conferences and awarded Best Scientist Award in 2015, Lal Bahadur Young Scientist Award in 2005, Science Award, Young Scientist Award in 2004, and Young Scientist Award in 2003. He is having a lot of research experience in the field of conservation of biological control. I would request to Dr. Muhammad Akram ji, please deliver his lecture in Hindi and English mix. Many students are attending this webinar here. If we will mix our Hindi and English language, so it will be more beneficial for our students also. May I invite Dr. Muhammad Akram sir?
Okay, good morning, sir. I am audible?
Yes, sir, audible, sir.
Good morning to all. Honorable Chief Guest, Professor Pramod Mal, sir. He is my senior, and then he is my table tennis colleague also at Pannagar and Shastri Bhavan. Professor Kamal Khiladi ji, he is my super senior. R.K. Mishra, he is my colleague. He is sitting behind me, just near with me right now. Dr. K. Shankar Ganesh ji, patron of this college and then this event, Professor Naresh Kumarji, Dr. Arun Kumar, a convener of this event, Dr. Dushyant Kumar, co-organizing secretary, and Dr. Johnny, organizing secretary of this event. Thank you for giving me this opportunity to speak with you and with your students today. So basically, I am not a person of biological control. Currently, I am working on plant viruses, and that too on the pulse crops. So I will be discussing on the general aspects, which will be covering plant viruses, management options, and then sustainable agriculture. So I'm just sharing my slides. So it is visible. Slides are visible? Hello?
Yes, sir. It is visible.
It is visible.
Yes, sir.
So I will be speaking because the time is limited. So I will not go much in detail. So I will be speaking on the what are the viruses, and then their importance, and then their management options,
Excuse me, sir.
and then
Sir, it is not under full view. You can make it full view under.
okay. So still it is not full view.
Still not. This side.
Okay. My slide is not full view, or
Not your slide is not full view.
This
just
Dr. Akram,
yeah.
it is bigger than the screen. So just minimize the percent of the slide.
Okay.
Yeah.
Okay. It's a really difficult task.
Sir, just you maximize it, na?
Yeah. Now it is visible.
It is visible.
Yes,
Yeah.
right.
It's okay.
So I will go back. Okay. So as we know that we all are dependent totally on the plants. Not only we, whatever the living creatures are there on the earth, we all are dependent on the plants. And then not only we depend on the plants for food, there are many other things we are dependent on them. So to save the plants is our prime duty. If you see the agriculture in past, it was very simple. The peoples were just gathering their food by just hunting, fishing, and all those things. So, but when the people have started to domesticate the plants and animals, this is called agriculture. And then after that, the population has continuously grown. And then at the same fashion, the agriculture also is continued to be grown. But why this agriculture has to come in the existence? It is because of the fashion or the need of the people there at that time, or it is just the response of the population growth. It is a matter of debate. But anyhow, today we are in the era of the agriculture. So in the past, the agriculture was not just organized, but right now it is organized. So after this Second World War, the world achieved the self-sufficiency in food. Or in other words, we may say that whatever the calories the world population needs, we are having enough. But the problem is that whether these all calories are distributed evenly or there is some mismatch. So even though that we are having the enough food production, but all population do not have the buying power. So the consumptions of the food are not distributed evenly among the population. Somewhere there is a large surplus of the food production and then availability, but somewhere it is a deficit. It may be at regional level, it may be a national level, or it may be a subnational level. So this is a kind of present agriculture. So whether we should go with the present agriculture or we need some modifications in that. If you see that self-sufficiency we achieved, so on what cost we achieve the self-sufficiency in food production? It is because of the increase of crop yield per unit of land or just expansion of the land. So whether the present agriculture is capable to meet out the demand of increasing population continuously? Yes, if we see that last 60 or 70 years history, we are continuously meeting out the demand. But at what cost? It is at the cost of pollution. pollution of air, pollution of water, pollution of soil, as a whole environment. So this is not a real agriculture. And then if it will be continued, then definitely there will be a huge loss to the humanity. So in this view that we need a different kind of agriculture, where we have to fulfill the nutritional demand, not just food, because we need to have to give the healthy food to everybody. So we have to have the nutritional demand fulfilled to the nutritional demand for the humanity in the world, and that too without degrading the environment. This is very important. So what would be the future agriculture? Because the present agriculture is just polluting our environment. If you see that the growth of the population is continue, and then we will be we will be reaching about 9 billion people on the earth in few years. So it is our duty that we have to provide the adequate nutrition for this population. And then not only just produce that much of amount, but the availability is very important for each and every human being, so that we have to enhance the buying capacity of the people also, so that everybody could get the available food. And then, or just to have a production of just equal to the demand, it will not solve the problem. So we have to produce a little bit higher, so that everybody can buy or access the food. So basically that the key issues are that whether the existing production can be used more efficiently, or whether the production can be increased without negative impacts on the global environment, or whether the present agriculture system can adapt to any environmental changes that do occur. या हमको कुछ और इसका अल्टरनेटिव सिस्टम चाहिए एग्रीकल्चर में, ये ज्यादा इंपॉर्टेंट है हमारे लिए. सो एक अभी हमारे जो चीफ गेस्ट हैं आज के डॉक्टर मल, उन्होंने कहा था कि वी नीड दैट सस्टेनेबल एग्रीकल्चर शुड बी देयर. Yes, there is a definition. I also agree with that. The sustainable agriculture is there. And then I'm not going to read this definition. But what we have to do in the sustainable agriculture, basically, that we need to satisfy the human food and fiber needs. And then we have to use the renewable sources more efficiently. And then we sustain the economic viability of the farm operation. Because most of the cases, the farmers are the sufferer. So that we have to enhance the quality of the farmers and the society as a whole. So if we do such type of things, that definitely we will achieve the sustainability in agriculture. There are ways that how we achieve the sustainable agriculture. Of course, that we have to diversify the agriculture. Yes, there are various options that maybe that some guest speakers will speak on that. And then there is a need to conserve the biodiversity, because India is a rich biodiversity country. So there is a need that we have to conserve it. And then we have to conserve our natural resources and then use judiciously. It is not just that कि हम दोहन करते चले जाएं अपने सारी natural resources का. हमको उसके अंदर बहुत judicious use करना पड़ेगा. Whatever the fertilizers and chemicals we are using, that should also be used judiciously. And then particularly for the management of the pests and diseases, so that we have to use the safer methods, safer chemicals, or the alternative methods. That is the topic of today's, that is the biological control. So we conserve these bioagents, and then we have to make efficient to use them against the pest or the diseases wherever they are available. If they are not available, then we have to make them available by doing the good quality of research. Now coming to the that my favorite part is that viruses. And then this is my favorite slide. I don't know how many still the people are believing that viruses are living or non-living, or they are in between. So my point is that nothing in between. The thing should be either the living or the non-living. So you have to decide yourself. But I would like to say one thing is here, that the virus is impacting very high on the humanity or the world as such. And then one case is you are just witnessing continue since from January to right now. Yeah, we are witnessing that how much losses or the how much disturbances a virus can do on the humanity. So I would like to quote here one शेर. ये मैं जब मैं बीएससी करता था अलीगढ़ मुस्लिम यूनिवर्सिटी में, तो मेरे फिजियोलॉजी के एक प्रोफेसर थे, वो हमको एक डेफिनेशन बताते थे एक शेर सुना करके. वो आज भी मुझे याद है. एक पोएट हुए हैं उर्दू के बहुत पुराने, ब्रिज नारायण चकबस जी. तो उस वक्त तो डीएनए की डिस्कवरी भी नहीं हुई थी और उस वक्त लाइफ के बारे में बड़ी अजीब अजीब टाइप की डेफिनेशंस थी. बट उन्होंने एक डेफिनेशन दी थी उर्दू में. उन्होंने कहा था, जिंदगी क्या है अनासिर का ज़हूर तरतीब, जिंदगी क्या है अनासिर का ज़हूर तरतीब, मौत क्या है इन्हीं अज्जा का परेशान होना. So what is life? So life is nothing. It's just orderly arrangement of the elements. And then you know the elements is the periodic table. Everybody knows that. So if you arrange orderly, that is the life. And then death, he says that inhi ajza ka pareshan hona. So disturbances in the molecules. So several elements, when orderly arranged, they make a molecules. And then if there is a disturbance in the molecule, this is the death. And this is very beautiful definition of life and death. So why I am telling this definition of life and death? Because now you can understand whether the virus is live or the dead. From my point of view, if you see the today's definition of life is the phenomenon associated with the self-replication of nucleic acid. And then you know the virus falls in this category. So from my point of view, that virus are living organisms. One can argue that outside the host, it is not multiplying. It means that they need the food. So every living organism needs the food. Without food, even no organisms can, even the human being also can't survive. So food is essential for everybody, for all the living organisms. That's why virus also need the foods. And then once they are having the food, so they are multiplying. So this argument is not, again, it's not applicable. So from my point of view, the virus is living, and then it is self-replicating molecules. And then if you see, the virus is very tiny. It has only two parts. One is the nucleic acid. It may be a DNA or the RNA, but never both. And it is having the capsid, the outer shell, the outer part of the body that is made up of capsid. And it replicates in the host cell, and it is carried from one plant to the other plant or one host to the other host by many vectors. I'm not going into detail all these because it will be a kind of lecture or student lecture. But just to refresh the everybody's that what is basically the virus is. And then this is again very important to understand the disease cycle. Disease cycle is a normal for fungal and bacterial. There are three things that should be there. That is the susceptible host, conducive environment, and pathogen. So if all these three things come together, there will be a disease. But in case of viruses, it is different. Why it is different? Because the fourth factor is also there, that is the vectors. And then vectors also should have the conducive environment, should have the susceptibility in the host, and they are also having many other effects on them. So for the virus, a triangle of the disease is not applicable as such. But why we have to understand it? Because we need to have to manage the disease. So we try to understand the weak point where we can hit, or which portion we can attack more to break the disease cycle, so that the ultimately benefit is there to control the disease. So here, while we are managing the viral diseases, so we have to think about the vector also, instead of the pathogen or the virus and the host and the environment, and of course the time. If you see the importance of the pathogen or the pest and pathogens, on the agriculture, it is very huge. It's around 2 lakh crore annual losses due to the pest and diseases and weeds. And then the loss is much more after the harvest because of these pests and pathogens. So it is very important that we have to have continuous efforts to save that much of losses due to pests and pathogens. So I'm just quickly going on to just refresh you some of the important diseases that everybody knows. There is the tobacco mosaic disease, it is in tobacco. And then windy yellow mosaic disease, which is everybody knows that we are having this disease. And similarly in the cucumber mosaic, watermelon mosaic, chili leaf curl. And then in in the chickpea, that is stunt, and mosaic disease in stunt. And then in pigeon pea, that is the yellow mosaic disease and sterility mosaic disease. Because of this sterility, you will not get even a single seed in the whole crop, in the whole field, if this disease has come. And another important disease in pulses, that is the yellow mosaic disease in urbean and mung bean, and then crinkle disease. And then it's very important virus that is called orthotoxovirus genus, and then the species is groundnut bud necrosis virus. It is causing disease in almost all crops in India. And then some of the photographs here that in urbean, and then French bean, and then pea, and then even potato, even tomato. So this virus is causing it's a very destructive disease. And then it is similar to sometimes it is similar to the fungal disease. So it is very difficult to distinguish it whether it is a viral disease or it is a fungal disease. So if we are not able to distinguish it, so for the management options will be very, very difficult to. Sometimes if we confuse that it is fungal and then we are spraying the fungicides, nothing will happen, and vice versa also. So just to refresh that there are many diseases of viruses. They are present, or they are in all agricultural crops and horticultural crops. But the other aspect is that all viruses are carried by some vectors, and these vectors are the tiny insects and then tiny molecules, not the bigger insects, especially the aphids, whitefly, thrips, leafhoppers, beetles, mites, and then even nematodes also. So they are transmitted in the field because of these vectors. So when we are thinking about how to manage it, so we have to think about these vectors. So if we efficiently manage the vectors, definitely the viral disease can be managed easily. So what are the options to manage the viral diseases? So there are two major targets. One is the virus and the other one is the vectors. So either we have to control the virus or we have to control the vectors. So for controlling the virus, so far we don't have any virucide. Still we don't have any virucides, especially to kill the virus in the host. Outside, virus can be killed easily, but inside the host, it is not. It is very difficult, rather than it is impossible. The reason is that because it is getting integrated into the genome of the host itself. So it is very difficult to have a contrasting chemicals to hit only the nucleic acid of the virus and then do not have any negative impact on the host DNA or host nucleic acid. Similarly at the protein level. So for controlling the viruses, so we have to develop or to breed the resistance cultivar, either through the conventional breeding or through the transgenic approach. That everybody knows that transgenics, the biotechnological approach that any foreign gene can be introduced into the crop plants or any plants. So they may have the ability to resist that particular virus. And then the other option is that we have to manipulate in the environment, like eradication of alternate host. You know that viruses also survive during the off season or when the crop is not there in the field on the alternate host. So we have to identify those alternate hosts and then we have to eradicate those alternate hosts so that this way the primary inoculum can be reduced. And the roguing is another important aspect for controlling the virus problems. And then sometimes the voluntary plants also carrying that particular virus. So we have to eradicate that voluntary plants also. Modification of cropping procedure also will help to reduce the incidence of the viral diseases. And then if sometime it is not possible in a particular area, so we have to go to the isolated area where we can easily manage the vectors so that the virus disease will be very less. And then the most important is that the virus-free seeds or planting material, we have to use it. So these are the options to manage the virus. And then the other option is there to manage the vectors. And then of course, the vectors are the aphids, whitefly, beetles, thrips. So they are very tiny, but they are very abundant in nature. They may be millions of the millions in the field. So for them, the peoples are using insecticides. And then we know that insecticide also creating the pollution in the environment. So we have to use the safer insecticides if at all is required, or we have to use the bioagents or botanicals, or even resistant cultivars against the vector also can be used. And then the trap crops. So I'm not going to much in detail about the biological control of these whitefly aphids. Probably Dr. Mal will elaborate what are the bioagents for these small insects, especially for practical point of view, whether they are there, some of them are not. But the safer chemicals are there. But if you use the safer chemical also, sometimes it is not again very useful. And the reason is that you are applying only in your field, but nearby field, the vectors are also there. And then even virus is also there. So, and then most of the safer molecules are the systemic in nature. So when you apply it, and then from nearby fields, if these vectors will come, and then first they have to feed on them, and then they are going to be killed. So meanwhile, they transmit the virus. So in most of the cases, if you are applying in isolated area or only in your field, any of the chemicals, it doesn't have much impact on the reduction of the viral diseases particularly, until you are not spraying in a larger area, so that the whole population of nearby that is going to be killed. And then after that, then transmission of the virus will be restricted. I'm going to tell you only one small success story of the transgenics. If I have to take more, then it will be a very lengthy lecture. So I have to finish basically in time. That's why, so this is one of the examples where in Hawaii, the whole papaya industry gone because of the papaya ring spot virus. So they have started to develop a transgenic. And then after developing the transgenic, they did, and then they trial it. And then they are very successful in developing this transgenics. And again, that industry revived. And then they are having now the transgenic papaya in the whole Hawaii. But this is island. And then doing such type of experimentation island is very easy. But in our country where diverse climatic conditions are there, it is really difficult task. So now I'm going to conclude my presentation. It is that what are the sustainable ways to manage the viral problems? Basically, that proper diagnosis of the causal agent is very important. And you know that government of India has already approved 3-4 bills related to the agriculture, and now the contract agriculture is coming into the country. So there the role of young agri graduate is very important. Now they have to train themselves to open the agri clinics, because now they will be acting as a like MBBS or the veterinary doctor is acting, and then they will playing very crucial role to bridge the gap between the farmers and then the industry people or the peoples who are coming for the contract farming. And then this will be the prime point where that every problem will be solved by the farmers as well as the contract, the company which will be having the contract to the farming. And of course, we continue do the research on developing the resistance cultivar, especially against viruses, because we don't have any viricides, or develop the transgenic crops using biotechnological approach. And of course, that these cultural practices are very important, that we have to focus on them. And then very little biopesticides are there for the viral problems, but whatever be available, that we have to use them judiciously. And then, yes, we can use the chemicals, but they should be safer and the need-based. It is not that any number of the chemicals we are going to use it. So finally, I would like to say that we have to exploit the resistant sources. And then for the vectors, we have to use the bioagents or biopesticides. And then, of course, that we have to conserve the natural fauna and flora so that these vectors can be eaten by the predators. We have to use the PGPR and biofertilizers to enhance the health of the crop. So if the crop will be healthy, definitely the disease will be very less. And then there is a need to continuously monitor the viruses, especially in response to the climate change. And then, of course, we have to adopt the technological advancement. And then whatever the challenges we are facing, that we have to face it and then take it as a challenge and then try to solve it. Thank you very much for patiently hearing me.
Thank you, Dr. Akram sir, for your valuable views, and just you enlightened on the future agriculture and viral disease in detail. Thank you very much. We wanted to listen more, but unfortunately we have limited time here.
So that's why I have taken the small presentation.
Thanks a lot, sir. I know you are principal scientist and you have a lot of research there, and you have a lot of views to spread among the students and faculty members. Thank you, Dr. Akram sir.
Okay, thank you.
We have another eminent scientist, Dr. Shankar Ganesh, who will speak on conservation biological control. Very sorry to say that we have very limited time here. I would like to request Dr. Shankar Ganesh, please try to conclude within 15 and 20 minutes. May I invite to Dr. Shankar Ganesh, sir?
Is it?
Thank you, sir. Thank you.
Yes, sir. Is it audible, sir? Yeah,
yes,
is
sir.
It audible?
Yes, sir. Audible, sir.
Yes, sir.
Hearable.
Yeah. Very good morning to everyone. Respected Chief Patron, Dr. Naresh Kumar, Principal of the college, and my Professor, Dr. Pramod Mal, sir, Chair of today's function, and my other colleagues from ICR, Dr. Akram, sir, Dr. Muhammad Akram, and Dr. Mishra from IAPR, and Professor Kamal Kladi, and Dr. Convener of this session, Professor Arun, and the organizing secretary of the event, Dr. John Kumar, and the co-organizing secretary of this event, Dushyant Kumar. Good morning, each and everyone. The topic which is given to me is conservation biological control and agriculture. As we all know, the process in response to population explosion. If we see in 2005, this population is going to double. Present population is going to double. So the population explosion, we have to increase the productions. So the current scenario of the agriculture is high input intensive agriculture. When we go for high input intensive agriculture, we have to go for high amount of application, high amount of places, pesticides to get the good amount of produce. So to keep the aim to push high amount of food production to meet production destruction of ecosystem. We are not considering the ecosystem in mind, but pesticides. Indirectly, it will affect the soil biota, which is more important. Spoiling the microorganisms. So, the natural enemies because we apply the pesticides, fungicides, herbicides, or whatever it may be, the whatever we apply or plant health or on plant, it is directly going to affect the trophic systems.
Sir, he might be disconnected. Oh, now he connected.
कनेक्शन में कुछ प्रॉब्लम आ रही है.
हाँ sir, there's 1 minute.
Yeah, is it okay now? Hello? Hello?
Yes sir, it is okay now.
Now you are
Yeah.
audible, sir.
So, yes, sir. Yes, sir. So, when we apply the indiscriminate use of insecticides and pesticides, it will affect the ecological balance in natural ecosystem. So, it will the pests which is present in the ecosystems will develop resistance, and the ecosystem will get spoiled through residue because of the pesticides, and the resurgence of the secondary pest, which is indirectly getting. So, the current scenario is urgently for harmonizing the deteriorating ecosystem by following agriculture, conservation agriculture. Yeah.
Arun sir, I think there is internet problem. You may call next speaker.
ठीक है.
Yeah. Sorry. Disconnected. So can I continue? Hello.
Hello, Dr. Shankar
Can I
Ganesh.
continue this?
Actually,
Yeah. Yes,
there is
sir.
a problem in internet somewhere. Connection problem.
Yes, sir. I'm continuing. If it is disconnected, then you can ask the other 2%.
Okay, okay.
Yeah. In cotton ecosystem, after the injection, I have to do after.
Dr. Pramod Mal, can I have Dr. Pramod Mal here? Dr. Pramod Mal. सुधीर जी देखें क्या डॉक्टर साहब है क्या?
Yes sir, he is connected with us.
अच्छा, Dr. Pramod sir, Pramod Mal sir, ex-head of the Department of Entomology, Govind Ballabh Pant University of Agriculture and Technology. He is a renowned scientist. He has published more than 47 national and international papers. He has published books and 9 book chapters. Dr. Pramod Mal is working on biological control. Now I invite Dr. Pramod Mal for his brief lecture and their views. Dr. Pramod Mal. Dr. Pramod Mal.
Yeah, I'm here.
Yeah.
Thank you, Dr. Arun Kumarji. Thanks for inviting me to deliver lecture. Please, some sound is coming from any. Please keep your microphone in mute condition.
सुधीर जी म्यूट कर दीजिए सभी को.
Yeah. So thanks to Dr. Akram. He was my batchmate, and He delivered such a nice lecture, and we also enriched by his good lecture on the viral diseases and their management in terms of eco-friendly methods of management. We could not hear completely. I think Dr. Shankar Ganesh could not complete his lecture due to some connectivity problem. So, but the topic was good, and he's very eminent scientist there, of course. So now I would like to start this presentation. I can share the slide, please. Professor Arun Kumar ji.
Yes, sir. You may share your slide. You are already permitted.
Okay, okay. Now it is visible to all.
Yes, sir. It is visible.
Okay, thank you. So this is a very important topic on which we are pondering.
Excuse me. Sorry for interrupting you.
Yeah, please.
Sir, can you make this slide in full view?
Yeah. Just 1 minute, please. I think there is some technical problem which is not.
Sir, okay, then you continue, no problem.
So I can continue this because it is not going to up to that extent. So this is, of course, we have a paucity of time, and I will try to brief in a few minutes. This conservation and biological control, biopesticide in agriculture. This is the very important for present scenario in our country, where although we are not using too much pesticide, but thing is that in some pockets you will find the maximum dosage of pesticides are being used, and in some pockets of the country there is no pesticide. So that is why this type of residue problem and so many other problems are there. And unfortunately, our friend insects, the pollinators and the enemies, natural enemies of the pest, are being killed due to this. So this is the matter of concern for us to think again and reorganize our this pest management strategy, so that we can have a safe environment and safe food. For pest management, the some they are being managed by using augmenting, rearing, and releasing bacteria, insects, fungus, nematodes, weeds, and viruses, and they are playing very important role in this pest management or sustainable agriculture. The food plants of the world are damaged by more than 10,000 species of insects and 30,000 species of weeds. Of course, 100,000 diseases caused by fungi, viruses, bacteria, and other microbes. As Dr. Akram said, that these are very important. In many of the crops, you will find thousands of diseases at one place, and this is really a serious problem in many of the area. and 1,000 species of nematodes are harmful for various crops. 33% of the crop loss are caused due to the insect pest. Now you can imagine if we can save these losses, then we can increase 33% production of the crop. Because the day by day, the land holdings are becoming smaller, the cultivable land are shrinking just, and the urbanization and so many other activities are there, which are just making the cultivable land dwindle. So this is problem. How we can increase or how we can bridge the gap of food requirement in coming years in 2050. So if there is no, we are almost reaching the maximum productivity of land. We can increase the production. Now it is in declining phase. The production is in declining phase, and we will have to think that what about the food requirement in 2050? Population is increasing in such a way that how we can just bridge this gap. This is matter of concern for all. And this only the insect pest and disease problems, if we can minimize, then of course we can increase the production 33% by saving this loss. Another scope is there. In many of the crops, the friend insects like pollinators are playing very important role, where the 30% increase is there. If proper pollination is done, or sufficient number of pollinators are incorporated in the field, we can increase average 33% of the production in that particular crop average. But in some crops like temperate fruits, if you are talking about the apple crop or peach, plum, and some other temperate fruits, in that crops the 7,000 to 8,000% increase can be achieved. 7,000 to 8,000. So in this way, the two ways, the entomology is going to play the roles in two ways. One is by saving the loss which is caused by the insect pest. Another is by increasing the productivity by incorporating sufficient number of pollinators in the field. So 66% or 60% around increase can be achieved. And this is only the way where the entomology has to play an immense role in meeting the requirement of the food grains in coming years. Question of food security. The world will need to produce 60% more food to feed an expanded world population by 2050. So this is the question of food security, where we are going to play an important role. As a plant protection scientist, they can play a very important role in this. And then, the three different phases of this agricultural pest control. The pesticide, we used the traditional approach, and then pesticides, and in 76 or 80s, we came to that IPM. Now in IPM, we are going to rectify you. Everybody knows about the IPM definition, that all the control measures are consolidated into a unified program in such a way that economic damage is avoided and adverse side effects are minimized. So this is the purpose of this. But now in this IPM, we are again going to rectify the things like the biological control. The role of biological control in this is more important. So this is, everybody knows that how the IPM base of the IPM is there. And these are the different tools of IPM. Then the sustainable integrated pest management, judicious use of pesticide, efficient utilization of natural enemies, conservation of natural resources, as the today's topic included the conservation words. So this is also going to play a very important role. Now we are aware about the ill effect of pesticides, so there is no need to discuss it again. Now this is important topic for us, the residue in foods. Now you can see many of the vegetables are the grains which we are using frequently. They are having a very high residue in that different vegetables. So if these vegetables we are eating, then of course they are causing damage to different vital organs of our body. So that is why now we are switching over to the eco-friendly management of pests so that we can avoid this lethal residual quantity or pesticides to save our health. If we talk about the conservation of this bioagents, the conservation biological control basically means keeping alive and enhancing the effectiveness of those natural enemies that are already present in that locality. The existing natural enemies must be protected or conserved, not even conserved, but promoted. And there should be some provision. Many conservation approaches are easily integrated into production regimen and can be very effective. Reduction of pesticide use is another important issue in that conservation of bioagents. The habitat manipulation for conservation of natural enemies. Many of the natural enemies, they are living in the various crops or in agro ecosystem. We will have to manipulate that agro ecosystem in such a way that it can become friendly to the natural enemies. How it can become friendly, if we are doing monoculture, suppose, and after harvesting of the crop, there is no plant the edge of the field or anywhere else. So this is the problem that the natural enemies are not having the sufficient food in that locality, and that is why their population is declining. If there is no crop, no natural enemies there. So some plants should be planted at the edge of the crop field, where the natural enemies can take their nectar, because most of the natural enemies from order Hymenoptera, and they are nectar and pollen feeder. So some ornamental plants or flowering plants should be planted, perennial plants should be planted that can render the nectar and pollen round the year so that the natural enemies can survive. So many plants are there, like the Cigalpinia is the plant that can be planted, or the coffee or the small edge plant that can be planted. Justicia is there. Some Tecoma trees are there which can be planted around the field. So these are the plants which can provide the nectar and pollen around the year to natural enemies. Alternate food source like this, you can see the buckwheat is there, and in some cases the phacelia is there. So these are the different crops that can be planted in between two lines of the crop or at the outer edge of the plant. Some the products like EnviroFeast on cotton attract to natural enemies. So some products are there that can be sprayed in the field for the survival of the natural enemies, and that can manage the pest population automatically. The non-crop vegetation, planting water chestnut in rice field, maintain the parasitoid Tetrasticus against the Serpophaga insatulus. So these are some examples, and many other examples are there which are not mentioned in that all cannot be included. I use an IPM program. The following IPM program is a great way to preserve, encourage natural enemies, identify the pest, learn its common natural enemies, and preserve it. These are the knowledge. This should be the farmers should be trained in such a way that they can get aware or acquainted with these friend natural enemies, and they can take action according to that. Now, this is everybody knows about the bioagents. These are some bioagents. The Coleoptera, the Coccinella beetle is there, which is very important biocontrol agents for many aphids especially, which is big problem in many of the crops. And some hymenopterans are there. which are playing very important role now. The Indian Mayna bird. This was the introduction of Mayna bird was made in 1762 to control the red locust, and then 1888 the first successful control of insect Katni kusani scale Iceria purchasei was made to control the Iceria purchasei to controlled by Rhodolia cardinalis. So these are the some successful attempts which were made to control the insect pest. Now you can see the exotic pests. Many of the pests we imported to control various important and destructive insect pests in many of the crops. These are the in India 166 exotic biocontrol agents have been introduced, of which 33 could not be released in the field. 6 provided excellent control, 7 provided substantial control, and 4 provided partial control. We cannot go far in detail. So you can see these some natural enemies. They are known to control a very destructive and devastating pest in many of the crops. This is the biological control, the microbial control, control, genetic improvement. This different components are there in this, and now we can see the non-pesticide control by the predators 41%, and then hyperparasitoids, parasites the 1%, and parasitoids 53%. So these are the some example by semiochemicals. You can see here the semiochemicals 15% genes by manipulating the genes 5%, and microorganism 30%, microorganism 34%. So these are the some data. Now you can see the world scenario where the percent of land under cultivation is 8% classical biological control. Where this classical biological control is being implemented, only 8% area is there, and augmentative biological control is 0.4%. So this is matter of concern, because we will have to switch over to that augmentative biological control and classical biological control both. Classical biological control is of course sustainable approach, while the augmentative biological control is just like insect control. Means this is not sustainable. Every time we will have to just rear the natural enemies and then release it in the field. So this is the various biological control activities. Now everybody knows about the trichogramma's importance. Because shortage of time, I cannot go one by one by reading all these slides. Then predators and parasitoids are two main biocontrol agents. Many of the predators are there from this Dictyoptera. You see the praying mantids and Coccinella. Parasitoids, Hymenopterans, they have the very good and efficient parasitoids. But the problem is that their reproductive capacity is very low. So like Coccinella, the reproduction rate of this predator is very low as compared to the aphids. As you know that aphids, they have they can produce the reproductive mode is very potential, like the pedogenesis there, parthenogenesis is there. So many they can immature stages directly can produce the offspring. So this is problem, and they are this Coccinella reproductive potential cannot be compared even with the. That is why this is not a success in case of the aphids in mustard crops especially. So important orders, because most of you are from entomology, so you are well aware about these orders. The beetles, you can see they are very important for us. A different species of this beetle is there in our country, and they are very potential predators for many of the natural pests. Then order Diptera, the civet fly, you can see this is one of the very important pollinator as well as the predator. So adult stages of this civet fly, can pollinate many of the crops. In mango crops, you know, this is the 80% of the pollination in plain area is performed by this important pollinators. And the immature stages, they are very good predator for the aphids. So they are very important again. And these are some robber fly, which are very good predator in many of the insects. The hemipterans bugs are there, and they are very lethal to many of the pests. But population is problem is that in this case, that the reproductive potential is very low. As you know that the hardly 15 to 30 or 40 eggs are laid by these predators, bugs, while the pest they can lay 200 to 1000 eggs in their lifespan. So this is problem that the we cannot rear up to that extent, we cannot augment to control or to release the sufficient number of predators required in the field. Then you see again the assassin bug, which is important predator. And then water striders is there, and then dragonflies everywhere. They are very prominent in many of the area where the ponds or the water sources are there. So this again the Neuroptera is playing important role in this. Insect parasitoids, you see the various families are there. Plenty of parasitoids are there in this. So now we can see some of the examples of these parasitoids. Some parasitoids are present in the Diptera, order Diptera. Technique fly, you see many of the pests, they are infested by this technique fly, and they kill the pest. So visual sign of this technique fly, you can see the parasitized pupa of pest, monarch butterfly. Hymenoptera is there. These are the important family which are playing very important role in managing the pest population or in biological control program. Now again parasitoid in Ichneumonidae. They are very specific parasitoids, and they are known to manage many of the population. Parasitoids from family Braconidae, you see these are the braconids, and we might have seen the braconid infestation in many of the insects host, which are very noxious to various crops. This is the Trichogramma. Everybody knows about Trichogramma and Trichogrammatidae family. This is very, very important family for many of the pests to control or to manage these important pests. So this is the Trichogramma life cycle. And these are the biological control approaches. Biocontrol agent augmentation, we can augment, and then we can inoculative release or inundative release, two type of release are there. And then biological control, we can conserve the natural enemies, or we can introduce classical biological control we can adopt. So we have already imported many of the natural enemies. Now you can see some of the successful classical examples of biological control in our country. The papaya mealybug, few years back, it was a big problem, but we successfully managed by bioagents a few years back. So this is again success rate of classical biological control agent of insect pest. So success rate, now you can see the Homoptera 68% is there. In other order, it is comparatively lesser as compared to Homoptera. So augmentation biological control basically means the adding natural enemies either where they are not present or present at a small number. Two different approaches to augmentation is there: inoculation or inundative release in the field. These are some important parasitoids, especially Trichogramma, which are playing important role in managing sugarcane pest, sugarcane borer complex. Biocontrol, biological control advantage is that it is self-perpetuating, pest specific, density dependent, cost effective, and environmentally compatible. And these are some constants because, in our country, the major problem in bioagents are application of IPM is that the illiteracy is the major problem in our country. The farmers are not trained properly. And that is why they are not aware. Even it is difficult for some institutions to apply in the field because a lot of knowledge is required to implement the biological control program. We will have to have the knowledge of about the biology and life cycle of these various hosts as well as pests, their natural enemies. So if we are not aware about their life cycle, then we cannot control the program or we cannot move with these successfully. This is the problem. So this is the future need of bioagents. Why we are just focusing on biocontrol agents. Now the microbial and sustainable IPM microbes. How these different microbes are playing important role? Fungus, protozoa, nematodes, and bacteria. Of course, viruses are there. They are very useful in this IPM program, and there is a need to emphasize these natural enemies to manage the pest population successfully. The bacteria, the bacillus and brevibacillus is there. I can see the shape, different types of bacteria is there, different genus is there, and they are very important for managing or applying the eco-friendly pest management in the field. Some fungus are there. Of course, Beauveria vesiana, Numeria, Metarhizium. These are the Licanium. These are the important fungus which can be utilized. We are hardly utilizing the potential of these microbes, and successfully there is a problem of formulation in our country that we are not able to produce the formulation of various these microbes. So that is why they are not available in the field. So of course they have a lot of potential, but there is need to utilize or evolve such a methods to have the proper formulation so that we can keep these for longer period of time and we can apply successfully. These are some viruses which are important, and the nematodes, of course, we are not having a sufficient number of formulations of these nematodes. These two nematodes are there, Heterorhabdis and Sternema. These two nematodes are very important for many of these soil pests like white grub, which is a very noxious pest of many of the crops. This is polyphagous pest, and they are very, very noxious in many of, especially in hilly area and the area where the upland is there. They are severe pest of many of the crops because it is polyphagous pest. So if we are able to just evolve the formulation of this nematode, then we can successfully manage this pest population. Now some protozoans, of course, they are lethal to many of the pests in many of the crops. So friends, this is the potential to cause pathogenicity in many insects. Now you can see the insect pests which are infected with these microbes, and they can produce a different type of symptoms. We are having the bacterial formulations. Now you see the death caused by bacteria. These symptoms are there. Now mode of action is there because sorry friends, I'm not able to describe each and every slides because of the shortage of time. This is the insect infected with the virus. There are more than 1,600 different viruses are there, which infect 1,100 species of insects and mites. Baculoviruses accounts for more than 10% of all insects pathogenic viruses. Mode of action of viruses there, then fungi, mode of action of this. Now you can see the insect infected with this various fungus or entomopathogenic fungus, which are very important for IPM program. And the nematodes are there. Now you can see the sternema emerging from Galleria larvae. We can just rear the Galleria wax moth, and that can be on that sternema can be reared to release in the field or to manage the pest. Then mode of action of protozoa, potential modifier of behavior. So friends, this now I would like to conclude my presentation because I have taken much of time.
Yeah.
So this is in, yeah, this in my view. If some sound is coming, please. This I would conclude that in today's scenario, there is a need to focus especially on the bioagents, biocontrol agents, and some microbes, which are very important, which can play a very important role. We are not harnessing even 5 to 10% of the potential available with them, but we will have to think it properly, and some more research are needed to have the appropriate formulation to be applied in the field, and so that we can manage the pest population successfully. So with these words, I would like to thank once again to all the organizers and patron, Principal Saab, and Dr. Arun Kumarji, Dr. Johnny, and other dignitaries. Most of them are well known to me, and most of them are from this GB Pant University. So this is really a proud moment for me. I'm able to see most of the persons are there. So thanks for this philanthropious work you have done today, and you are doing. And thanks for just giving me opportunity to share my view with you, great people, venerable dignitaries, and all the participant students. If anything is there, any query, if you are having, then you put it forward for discussion. If anybody interested, please, you can put your query forward discussion. Thank you. Now
Thank you, Dr. Mal, for your wonderful lecture. Definitely, you delivered a very nice lecture on conservation of biological control. And we would like to continue this lecture series in future also. And we would like to request you to another lecture whenever you will have time for us. Thank you, Dr. Mal, sir. You just gave me your precious time for all of us in this.
Thank you. Thanks.
Thank you, Doctor. Now, series of this lecture, we have another eminent personality here, Dr. R.K. Mishra. He is a principal scientist at Division of Plant Protection, Indian Institute of Pulse Research, Kanpur. He has a lot of experience on host plant resistance in molecular plant pathology. He has identified the isolates of different trichoderma species. He has a vast experience in area of plant pathology and control of plant disease management. It's a great honor to tell me here, Dr. R.K. Mishra has published more than 100 such papers and conference papers in different journals. He has got a Young Scientist Award in 2017, and he has published 5 books in his name. I think Dr. Mishra ji is with us. Dr. Sudhi ji, Dr. Mishra sir hai?
Yes, sir.
May I invite Dr. R.K. Mishra sir for wonderful your lecture here. Very sorry to say here that we have very short period. I can understand that you have a lot of experience. Dr. R.K. Mishra sir, please go ahead.
Yes, sir.
Dr. R.K. Mishra, sir, your face is not visible. Can you adjust your camera?
Yeah,
please.
Visible, sir?
Yes, sir. No, your slide is visible.
Slide is visible.
I am
Face
not
is...
visible.
You are not visible.
Face is not visible. Face is not visible.
I think, sir, you will have to adjust your camera, sir.
This is not visible. Visible, sir, or not?
Not visible. Not visible.
Sir, your slide is visible, but unfortunately, so your face is not. I think there is a problem in camera.
So first you just you don't share this slide, then it will show your face.
Stop sharing, sir.
Now just you are not visible, that's up. Your only the hairs are visible. You please come in front of camera.
Visible, sir.
Yeah,
Yes, sir.
you little bit you can yeah now you now it's okay.
Okay, okay.
Okay, you can start it.
Please start, sir.
My PPT visible, sir. I am audible, sir.
Audible, sir. Audible.
आवाज आ रही ना सर? Okay, sir. Good afternoon to all my respected seniors, friends, and dear students. First, I am highly grateful to the organizers to invite me as resource person to share something on biocontrol aspects, what we are doing in IIPR in pulses. So, just my topic of presentation is conservation and characterization of potential biocontrol agents from pulses rhizosphere for effective management of soil-borne pathogen of pulses, especially in wilt of chickpea, pigeon pea, and lentils. You know that wilt is the major problems in pulses. More than 20-25% losses nowadays. डॉक्टर मल साहब ने तो सारी चीजें बता ही दिया जो है डिटेल about the biocontrol agents and their conservations. Just be focused only on pulses. What we are doing in pulses for management of the soil-borne and root rot disease. Just the rationale, you know that pulses are the most important grain legume crops grown under arid and semi-arid conditions worldwide, and they are very rich in dietary proteins. You know that without pulses, nothing. And it increases in fertility of soil as well as microbial diversity and in their rhizospheres. But nowadays, very less exploitations of especially in microbial rhizosphere microbes from pulses rhizospheres. So there is a vast array of potentially useful of beneficial microbes still habitats. And you know that about 15 to 20% annual yield loss in pulses due to the several biotic and abiotic stresses. Among the biotic stresses, soil-borne is the most potential threats causing every year. So being a soil-borne in nature, fungicides are not economical, viable, and feasible for the management of these soil-borne diseases. So the biocontrol is the most economical and viable options for management of these impulses. These are the some important soil-borne disease and their symptoms. Then you know that the biocontrol agents that Malsa has described details. Biocontrol agents is a method for controlling the pest and disease and nematodes through the another living organisms like the antagonist, which is the beneficial in nature. Just some why we are using the biocontrol agents. These are the some success of biocontrol agents and their formulations. These are the suiting to different agroecological niches. These are the longer shelf life easy mass multiplication and their applications in different crops, and also ability to tolerate the different agrochemicals and compatible to other beneficial microbes like PGPRs, and ability to multiply on infection sites. And these are the broad-based efficacy against the major phytopathogens. So these are the some success of the biocontrol agents. So we are focusing to use biocontrol agents. These are the some important, the major, most important biocontrol agents generally we are using. Like if we are talking the fungus, अगर हम फंगस की बात करते हैं तो ट्राइकोडर्मा, जितने भी हम बायोकंट्रोल यूज कर रहे हैं नाउ ए डेज, मोर देन 60% जो यूज हो रहा है वो ट्राइकोडर्मा उसमें कवर कर रहा है. उसके बाद आपका पीजीपीआर, Pseudomonas bacillus, Rhizobium, and ENPV is the viruses using as a biocontrol agents. So these are the important biocontrol agents people are using. This is our study. We collect the data from the Google Scholars, the scenario of biocontrol agents research in different crops worldwide, including patent publications. You see the trend biocontrol agents, it is going in increasing order. These are the red indicated with the whole total crop plants, biocontrol agents in total crops. We are just be focusing the scope of biocontrol agents in pulse crops. So you see the lot of scopes in pulse crop. It's also going in increasing orders. These are the 15 years data we have compiled from the Google Scholars. In biocontrols, most of the people are using in trichoderma. Most of the research is going on in trichoderma aspects, 60% say. So why we are using in trichoderma? These are the lot of several benefits, the use of biocontrol agents. These are the importance. These are also having the plant growth promoting activity, having the bioremediation activity. These are the some strains of the trichoderma having the industrial use. Then come to our research at IIPR, what we are doing at IIPR in the field of trichoderma and biocontrol agents. So nowadays we are having the more than 100 trichoderma isolates belonging to 8 different species, like the Trichoderma harzianum, Aspergillum, Langibrachiatum, Verens, Atruvinium, Aspergillus, and Afroharzian. 8 different species, and isolated and identified and characterized on the basis of their several characters: morphological, pathological, biochemical, as well as molecular characterizations. These are you see the diversity of trichoderma. We have collected all over the country from the major pulse growing areas. These are some morphological diversity you see among the different species. We have also the characterized on the basis of molecular characterization by using the ITS as well as test primers. And we have all these isolates we have tested against all 3 built pathogens, pigeon pea, chickpea, and lentils. Out of 122 isolates, we found that 2 strains like IIPR TH31, IIPR TH33, these 2 isolates having the most potential to inhibit the growth of the 3 built pathogens. We have, and as well as also these 2 isolates having the plant growth promotion activity also, and having the high chitinase activities. So these are 2 isolates are having the multi-trait like antagonistic, plant growth promotion, and some biochemical activities also. So you see the how their potentiality of the these trichoderma. I mean, under in vitro conditions. Besides trichoderma, we have also characterized some PGPR isolates identified from the pulsar rhizospheres. More than 100 isolates of the PGPRs belonging to Pseudomonas bacillus and some other species, and well characterized on the basis of morphological and their plant growth promoting test, as well as biochemical test. And these are the tested against in vitro activity and antagonistic activities. Out of these, we found some PGPR isolates having the potential. Nowadays we have having the two type of formulations, talcum based as well as formulation granules based. So on the basis of this, we have developed two formulations in the name of Dalanderma, because I am working in Pulse APR, Pulse Institute. So we thought we develop a product in the form of in the name of Dalanderma. So Dalanderma and then Dalanderma 1, the potential strains. And these two formulations we have evaluating in pot conditions as well as in field conditions. These are the we develop a methodology how we proceed, how we prepare and develop these formulations in lab conditions. And we are using in the two modes, seed treatments as well as soil applications and farmer fields. These are the some pictures for the use of trichoderma in the pot conditions as well as in field conditions. You see the if we are using in trichoderma in field conditions for the managing the yields, inhibit the root rot and build pathogens in lentil chickpea, and it decrease the disease levels, and also increase the plant growth promotion activity, also increase the number of nodules and root bed and shoot bed, and also total biomass of the plants also. And some biocontrol formulations is available, you know that all that. Now come to the conclusions for the just to increase the improve the efficacy of the biocontrol agents for the management of phytopathogens. We need to develop a host and pathogen specific biocontrol agents and their formulations, and we also focus to develop a multi-trait biocontrol agent formulations having the different trait like antagonistic, plant growth promotion ability, biochemical, high chitinase and high cytophores. So we develop a multi-trait biocontrol formulations can be used as a formulation as well as consortia for holistic crop and soil health management in pulses. So nowadays we have a formulations in the name of Dalanderma, and we have having the more than 100 isolates of trichoderma as well as PGPRs, especially for the from the pulses rhizospheres. So these are the conclusions.
Thank you, Dr. R.K. Mishra ji. Hello.
Okay, thank you, sir. Yeah, sir.
Thank you. Thank you, Dr. R.K. Mishra ji. In a very limited and very short period, you just told each and everything about the biocontrol agents, trichoderma, and especially Dalanderma.
Yeah, I will go. We are working in pulses, so we focus on.
I will go through this your research work and definitely spread the knowledge among the students. So thank you, Dr. R.K. Mishra ji. Our next speaker is Dr. Kamal Khiladi. He is professor at Sardar Vallabhbhai Patel University Department of Plant Pathology. He is very good friend of mine. Dr. Kamal Khiladi, he is specialized in biocontrol.
Excuse me, sir. He is not connected with us.
दुष्यंत जी, डॉक्टर कमल खिलाड़ी से बात करें. कमल खिलाड़ी. Dr. Dushyant is trying to talk to Dr. Kamal Khiladi ji. I think Dr. Shankar Ganesh ji is here. Shankar Ganesh ji.
1 minute.
Yes,
hello.
Dr. Shankar Ganesh ji, are you hearing me?
Yes, sir.
Your connectivity is correct now?
Yeah, it is getting right.
So, can I continue with you, sir?
Yes, sir. Can I continue?
Yeah, Dr. Shankar Ganesh ji, actually in between the internet problem was there. That is why I asked to Dr. Shankar Ganesh ji, please continue your lecture here. Dr. Shankar Ganesh ji.
Yes, sir.
Yeah, Dr. Shankar Ganesh, please go ahead.
Good afternoon to one and all. Sorry for the interruption. Due to low internet connectivity, I was not able to continue the last session. Sorry for the interruption. So, as we all know, the population explosion, indirectly we need the more amount of food. When we grow, when with the more amount of food means we have to cultivate more crops. The more crops, when we are more crops with less land, we have to go for intensive agriculture. Intensive agriculture, it requires a high amount of fertilizers, application of high amount of fertilizers, pesticides, without considering the any ecological systems. So when we applying the high amount of fertilizers, it will affect the soil health, it will affect the entire biota. So entire biota means it includes natural enemies associated with the second, third, and fourth traffic levels, ecological balance through application of high toxic pesticides. It indirectly leads to resistant development in the target pest, residue in the cultivated crops, resurgence of the secondary pests. So these are all the things when we are not considering the ecosystems in mind. So our aim is to urgent need for harmonizing the deteriorating agro ecosystems. So since I am working in cotton, the last 20 years, cotton pest scenario has changed. After the introduction of BT cotton, before introduction of BT cotton 2002, the scenario of pesticide consumption was towards application of insecticide for controlling bollworms. After the introduction of BT cotton from 2002, the sucking pest complex which are present in the cotton ecosystems, which has emerged as a major pest. From 2005 to 2010, the Phenacoccus vulnus is a major the sucking pest. It's a mealybug which has caused huge loss to the cotton crops, almost all 10 cotton growing states of India. This pest is introduced from Pakistan, from South America to Pakistan, and later it was come to Punjab and it has distributed all 10 cotton growing states. Then this pest is having more than 154 plant species, plant to 53 families. And the one peculiar thing is, along with the pest, when this pest was introduced to the natural to the new area, along with the pest, the particular natural enemy, that is Anaceus aeruginoensis, is a wonderful natural enemy. Also, that is called pernicious biological control. When the pest is when the parasite is introduced along with the pest, this Anaceus aeruginoensis, this parasite got established wonderfully. If we are not spraying in cotton ecosystems, this pest can manage very easily. But unfortunately, farmers are not considering this effect, the beneficial nature of this parasite. They are blindly broadcasting the pesticides like anything. From 2008 to 2011, another important pest that is also mealybug, Paracoccus marginatus, it has caused huge loss to the not only cotton, ornamental plants, mulberry industries. It has silk cotton. This pest has caused huge loss, not only cotton, papaya, and other economically important crops. To manage this pest, our NBR, the ICR NBR, has brought 3 parasites, Acinetobacter papayae, and another 2 parasites from USA. These parasites are belongs to Encyclidae family, and this Acinetobacter papayae got established very well under present situations. Then switching out from 2015, the whitefly problem in Punjab has created huge loss to the cotton farmers, particularly Punjab, Haryana, Rajasthan, and those areas where the this pest, the whitefly has not only caused a sucking pest problem, it has transmitted cotton leaf-fly virus. So whitefly outbreak in Punjab, Haryana, and Rajasthan during 2015 mainly due to large-scale cultivation of BT cotton, which are highly susceptible to whiteflies and the cotton leaf-curl virus. Excessive and indiscriminate use of piperonal, one of the most notorious insecticide, and along with the mixture of the tank mixture of pyrethroid and acipates. These are all the main causes of a main problem to outbreak of the whitefly. From 2005 to 2010, another in South India, green bug, that is merit green bug, caused, especially in Karnataka and Tamil Nadu, this emerging pest. Similarly, in tobacco caterpillar Spodopreta. So in cotton crop itself, from 2000 to 2020, there are n number of pests emerging and creating huge loss to the cotton growers. Only cotton. The cotton is an important crop which is consuming more than 50% of the pesticide consumption. So the next come to this Spodoptera frugiferida, which has caused huge loss to the maize crops all over India. Now this is a huge, this is a major issue in agriculture ministry. And another one is the desert locust. This is the emerging pest in India, which is causing huge loss. How to manage? This is alarming situation. How to manage this? Every day there are new pests are coming, and how to manage this? Why this kind of huge and Q and cry, this so much problem? All the above problems are arising due to poor ecological consideration in pest management. When there is a ecological balance, nothing can come as a new pest. We are not considering the ecological systems. Without considering the ecological balance, we are applying the pesticides. So the very important thing is biological control. Why biological control? Biological control is self-sustaining in pest management. With the except biology control, no other method can sustain the ecological balance, because biology control regulate the pest population by the activity of natural enemies like predator, parasites, and pathogens. These are basic things. Augmentative biology control. One is inundative, another one is inoculative. In inundative, we know how we are releasing the periodic release. Inoculative means we are releasing the parasites and we are leaving the F1 to establish under undisturbed ecosystem. In case of classical biology control, we have to release the biological control agents for controlling introduced pest or for native pest. Periodic we have to release. The best example is from India, we have exported miner to control red locust in Mauritius. This first classical biology control. Status of conservation biology control in present day agriculture. Because when we are applying the pesticide continuously, we are not considering the ecosystems. We are continuously deteriorating the natural ecosystem. Complete removal of the plants after harvesting gives rise to disappearance of the natural enemies after each crop season. This we can see that when we are harvesting the harvesting the crop, sugarcane like crops, we are just removing the crop and we are burning the stubbles, and whatever the best example, Pyrilla perpusula is a pest. Because of the burning of stubbles, all the natural enemies which are associated with this sugarcane ecosystem getting disturbed. What is conservation biological control? Conservation biology is nothing but protection of natural enemies against adverse effect of pesticides and incompatible cultural practices and improving their efficacy by providing food sources. Just without any disturbance, you just leave the natural enemies to establish so that it can sustain, the F1 can sustain, and it can easily manage the pest, whatever it comes, either sucking pest or lepidopterans or whatever it may be. The more efficient conservation biological control involves three main practices. One is collection, then another one is preservation and release. The collection of natural enemies. When we have to collect the natural enemies, we have to collect the natural enemies from the fields shortly before without complete removal of the plants and disappearance of the occurring natural enemies. When you see the end of the cropping season, the natural enemy population will be high and the pest density will be low. So by that time we have to harvest the natural enemies. So there are different seasons of their summer collection. Why we have to collect the summer collection? We have to collect the natural enemies summer, because in summer there will be harsh weather. So the harsh weather situations, the natural enemy can't sustain. The natural enemy will not get any sufficient food. So similarly, winter collection we have to do during winter. The natural enemy may not be getting sufficient niche to sustain. Similarly, monsoon season also, the natural enemy balance will get disturbed. So end of crop season, host density decreases, natural enemy strength increases. This is the ideal time to collect. So collection techniques, there are different collection techniques. Picking up the infested leaves. When we pick the infested leaves along with the host insect, there may be chances of natural enemies inside the pest. So we can collect the natural enemies, just infested plus sweets, and beating the branches in and cloth bags, just to be infested plants we can collect through sweeping net. Mostly parasites are collected through sweeping nets, chrysophids, syrphids, coccinelle species. This is particularly if you see during winter season, particularly mustard growing time, there are a lot of chrysophids, coccinelles and chrysophids and syrphids. Syrphids and coccinelles are the plenty if you can, if you, particularly in flowering time of the mustard. If you go to the field, you will get enough number of syrphids and coccinelles. Through if you can collect and you can preserve it in the lab condition, under you can establish under lab condition and use it whenever you need it. Direct collection of individuals. The parasites catch caterpillars like Helicoverpa, there are n number of endoparasites, larval egg parasites, larval parasites, which are present in the undisturbed ecosystem, can be collected and it can be established under lab conditions. Through aspirated devices, these are simple techniques for collecting the flying natural enemies in orchards or vegetables or any ecosystems. Okay. First, we have collected and we can establish under lab conditions. Preservation of natural enemies. Many plants can provide food sources for natural enemies like nectar and pollen and plant shape. Because the natural enemies, adults, mostly adults of the natural enemies, I'm talking general, in general parasites, adults of the parasites, they're mostly they are phytopagus. Phytopagus means their main survival is depends on the pollen and nectars which are present in the host plants. When we allow the specialist natural enemies reproduce only in the presence of their specific prey host, generalist predators can only multiply, generalist predators can multiply whatever the available host, but specific natural enemies only multiply with a specific host. Plants that produce a lot of pollen like resinous, that is castor plants, and provide more pollen for predatory mites. Not only mites, these castor plants producing pollen will give more. It is a very good plant for either parasites or predators, including pests. Food spray, there is when the non-availability of these naturally occurring the either plant pollen or nectars, we can go for artificial or natural food supplements can be sprayed or dusted onto the crop to suppress the natural enemies in crops, where nectar and pollen are absent or only present in low quantities. So this kind of, whenever there is a low density, we can go for artificial spray. For example, pollen spray can serve as a food for predatory mites. Predatory mites, which are very important in case of white flies and cucumber. Applying artificial food for natural enemies. Application of yeast and sugar in chrysanthemum, which will attract the astigmatic mite, which are spray for the phytosil mite. These phytosil mites are the very important predator for the astigmatic mite in most of the crops. Artificial field rearing units. We can establish the rearing unit under field conditions for controlling the saprophytic fungi. Supplementing mixed diet for natural enemies. We can, for example, supplementing thrips with pollen increase the egg production of Aureus laevicatus predation rates by of thrips larvae. The Aureus laevicatus is a predator. Providing oviposition site. Whenever there is an insufficient natural niche, we can provide the oviposition site for natural enemies, suitable oviposition site for essential for reproduction of many predators. The hard plants which will inhibit the establishment of the predators. So when we provide the soft host, it can establish under natural conditions. Similarly, when we harvest the tomato, de-leafing practice is most common. This has a strong negative correlation for establishing the natural enemies, which are associated with the leaf miner or any tomatoes, tomato fruit feeders. Planting suitable non-crop plants near fields. When we planting the non-crop plants, which will harbor the natural enemies associated with the crop pest. So it will indirectly help in the enhancing the population strength, natural enemy population strength. Inducing the plant responses through application of odors. Preservation of natural enemies might be engaged in differential crops by breeding varieties that produce more volatiles and nectars. When we breed the plant, this is a breeding process. When we breed the plant, which can give more plant volatiles and more nectars, which can attract the insect pests along with the natural enemies, will definitely enhance the ecological balance. Applying semiochemicals, one of the important method, because it is a costly affair, but applying semiochemical which are similar to the host insect will enhance the establishment of natural enemies associated with the host insects. To minimize the application of insecticides or through keeping the consideration of the existing natural enemy in the cropping systems, selecting the pesticides which will help to establish the natural enemies. So first we have collected and we established and we have to release the natural enemies. There are different techniques are there in the releasing techniques are there. Egg parasites, for example, egg parasites, larval parasites, people parasites, and there are different type of parasites are there. Timing of application of different parasites are there, and even for pathogen also there are different techniques are there. For example, application of egg parasites. We are going, if we apply, if we want to release the egg parasites, we have to have parasitized eggs in the egg cards. Through egg cards, parasitized egg cards. For example, rice tempora. Corsera moth is mainly as a main host for the establishment of Trichogramma chironis, Trichogramma japonicum. Similarly, Galeria melonella is an important larvae for establishment of the endomopathogenic nematodes. So when we go for application of trichogrammatids, application of trichogrammatids once in 30 days, Trichogramma chilonis and Trichogramma japonicum will control the rice stem borer as well as rice leaf folder. Similarly, sugarcane stem borer at the rate of 1,50,000 once in 3 months, which will enhance the establishment of Trichogramma chilonis and Trichogramma japonicum. Similarly, cotton bollworms. For in case of Helicobacter pharyngeata, Trichogramma chilonis was very good parasites. In case of pink bollworm, Trichogramma bacteria, which is important parasites. These parasites can be established under lab conditions. It can be applied periodically. We can apply and we can minimize the pest load. The larval parasite, important larval parasite, Bracon species are there. So very important externally parasites which can manage the pest very easily. But thing is, we are not considering the ecosystems. Whitefly encarsia is a major. When there was a outbreak of 2015, when there was an outbreak of whitefly in Punjab, all entomologists focus was where is the encarsia sparmosa, which is infesting the whitefly, because application of piperonil and tank mix of acephate and with piperonil and pyrethroids, which killed the encarsia sparmosa in natural ecosystem. That's the main reason whitefly parasites could not establish and outbreak has occurred. The aphid parasites, as I already said, aphid parasites, Liphophis, we could easily know in Liphophis are important aphids which cause huge loss to the mustard. Aphid parasites, there are more number of Apidia species are there, and predators are there. Coccinelle predators, Coxella septum punctata, kilo minus 6 maculata, and similar. There are 8 predators are associated with the aphid ecosystems. The parasites of scale insects, mealybugs of parasites are already said, Acerobacus papaye, Anaceus argonensis, Anagyrus loci, Leptomastix lactulopi. There are different parasites. We can name it. Then predaceous mite, which can easily control the soft-bodied insects as well as lepidopteran insect pests, which are associated with the major crops of. Now we have seen the collection, the preservation and release. The conservation, how to conserve in brief. Population of natural enemies subjected to continuous deterioration of the modern agriculture practices. This can be minimized by avoiding the deteriorating practices. During non-crop period, we have to give the niche to establish the natural enemies. To preserve the natural enemies can be achieved by providing habitat and resources for natural enemies. Collection should be conducted before crop harvest, but also could be done during the crop growing season and during winter and fruit orchard and in permanent ecosystems. Preservation of natural enemy very important, vary according to the types of natural enemies and target pests and plants and ecological conditions. Many plants can provide food sources for natural enemies like nectar, pollen, and plant sap. Whenever we remove the adjacent host, we have to keep in mind whether this adjacent plant is really it is a alternate host for the pest, or whether to remove or not. We have to keep in mind. Otherwise, that adjacent crop may be a better host for the natural enemies to establish. By giving artificial natural food supplement for the whenever there is a shortage of natural foods. Introducing plant harboring the prey species is essential for the preservation of natural enemies. Plant harboring the prey species. For example, whenever we want to establish the phytocid mite, that is phytocid predatory mite, we have to establish the phytocid mite. In case of chrysanthemum, yeast along with the sugar in chrysanthemum, we can easily enhance the predatory mites. Releasing techniques also vary with the crop stage and the type of the pest and type of natural enemies. There are different, these techniques already explained. Conservation biology in plant pathogen, one better example is use of organic mulches. Reduces the graphene Botrytis this pathogens for this primary inoculum. By the way, the biological system is potentially applicable to other pathogen also. So this is one better example to for the conservation biological control of the pest. The other one is biological control is bio-pesticides and bio-fungicides. These bio-pesticides are nothing but managing the invertebrate pests through plant pathogens and weeds, and microorganisms such as fungi, bacteria, and viruses. These are the major bio-pesticides which are being used to control the micro insects. Similarly, biofungicides attack plant pathogens through several mechanisms, including antibiotic and hyperparasitism. Many agents are highly competitive for space and nutrients, but usually have low capability for displacing the developing microorganism. This is one lacuna why biofungicides are not that much successful as compared to the insecticides. The conservation agriculture in respect to pest management, if you look at shelter belts, winter breaks, intercrops, and polyculture techniques, crop rotation, cover crop, and organic soil maintenance, these are all the techniques which can increase the natural enemy species diversity and lower the pest population density. If you see the lower part, conventional tillage, if you follow the conventional tillage, it will decrease the natural enemy. monoculture, if you follow the monoculture techniques, naturally, there is no chance for establishing the natural enemies. Total weed removal, if you are not considering the ecosystem, whether this weed is the alternative host for the existing pest or not, without thinking those in that point of view, it is definitely it is a futile exercise. Application of chemical fertilizers without knowing the necessity of the crops, necessity of the environment, without considering the ecosystem, broadcasting the chemical fertilizers will directly it will affect the establishment of the proper ecosystem. Similarly, pesticides related effects, direct effects, we have already seen. These are a simple example. Intercropping peanut, coriander, maize, soybean helpful in the enhancing the activity of beneficial predators in any ecosystems. Similarly, coriander is a better crop for the establishment of the natural enemies. There are some prepared plants for prepared pests. Those prepared plants and prepared pests is a better host for the natural enemies. Like marigold attract fruit borer pest. Similarly, the growing of these plants in main crops, along with the main crops in chilies, will reduce the fruit borer damage in the crops. Similarly, soil nematodes, which are harboring in the vegetable ecosystem, which can eat easily, we can minimize by growing the marigold plant. Similarly, growing castor plants along with the chilies or cotton or tomato, castor is, as I already said, castor is an important important plant which can harbor the more number of natural enemies because it has good pollen source. Will reduce the fruit borer, Helicobacter parmigiana and Spodopterytra, because Spodopterytra, it is a castor is an important preferred host for laying eggs, and even in lab condition also Spodopterytra is easily established under with the help of castor plants. Similarly, trap crop can be identified and cultivated to reduce the pest infestation. These are the NCEPM National Center for Integrated Pest recommendations. Cotton plus soybean, cotton plus cowpea, cotton plus pulses, cotton plus groundnut will easily we can minimize the sucking pest problem in southern zones. Intercropping or trap cropping of cottons, like use of trap crops like okra, cannabis, castor, marigold, early pigeon pea, jowar, maize is recommended in cotton ecosystems. Insects feeding on those crops can be removed and destroyed because those are trap crops. And however, in northern zone, okra should not be used under cotton and should not be grown in and around orchards because the okra is a major alternative host for the whiteflies. Intercrop as a cultural management in cotton, because in Bihar carry caterpillar is a planting castor, because castor is an important plant which can harbor the Spodopterytra, Bihar hairy caterpillars, the insect pest, any hairy caterpillars which can easily harbor the castor. So by planting the trap crops like castor, we can attract the insect pest on trap crops, and by the way we can save our host plants. For whitefly management, cultivate alternate inter-host crops such as tomato and castor and the boundaries to trap and destroy them. For thrips management, grow cowpea, onion, soybean as an intercrop in cotton to reduce early stage of the pest. In American bollworm, trap crop with like tomato and destroy them when the population is high is an important practice followed for cotton. Use of maize and cowpea on borders and wild brinjal and cetera as an intercrop significantly help in reducing the pest population in cotton because these are the best source for the insect pest. Intercropping in gram. In gram, use of African marigold as a trap crop, which will harbor the natural enemies of the pod borer, which will easily manage the bollworms, pod borers. Intercropping in pigeon pea with sorghum for reducing the Helicobacter wilt and nematode incidents. Intercropping in groundnut, which will like pigeon pea, pearl millet, 3 to 6 hours groundnut, and 1 drop intercrop, enhance the activity of pernicious, this is a parasite. Grow trap crops like cowpea, castor, and jatropha on field buns to attack the red-haired caterpillars. When we attack the red-haired caterpillar, we can easily destroy them in the border crops. Intercropping and mixed cropping practices followed by recommended by ICRISAT, pearl millet along with the cluster bean, pearl millet with cowpea. These are the important practices in different zones which are recommended by ICRISAT. In conclusion, conservation biology control and biopesticides have a great potential in organic agriculture or in IPM programs. These systems are complementary and elements of them can be integrated into conventional pest management systems. There are two techniques. One is conservation and conservation, another one is preservation. These are the two techniques do have their limitation. However, and most agriculturists need to adjust their perception of the technologies, especially on biopesticides. Agriculturists require education about the biology of the biopesticides and the target pests, because biopesticides have to establish under natural ecosystem. It is having its own limitations. However, it's having own merit also. So we ourselves have to get educated. Then only the farmers will get benefited. Yeah. Acceptance of the limitations and the gaining of appropriate knowledge should allow biopesticides and conservation of biological control techniques to be integrated into agro ecosystem to produce a more sustainably produced higher value product. When we keep the negative impact of these biopesticides and parasites in aside, and when we allow the parasites to establish, it's a wonderful high value crop, and it's a wonderful techniques which will be helpful for the future generation to save and to live healthily. This approach can enhance the contribution of ecosystem services to pest, weed, and disease control, satisfying increasing environment, energy, human health, and marketing demand. So as long as the conventional pesticides remain relatively inexpensive, these economics of these biopesticides and natural enemies cannot be understood. So we have to understand both conventional and biopesticides, positive and negative. And by the way, we have to choose which is more which is more suitable for our future generation. It's our view or our thinking to change, to adapt, whether we have to go for conservation or we have to go for conventional. Thank you so much.
Thank you so much. Thank you very much, Dr. K. Shankar Ganesh, of your valuable suggestions and valuable lecture and wonderful lecture on conservation of biological control. Actually, we had Dr. Kamal Khiladi in the series of this webinar lecture, but unfortunately, due to connectivity failure of Dr. Kamal Khiladi's phone or somewhere problem in connectivity, he's not here to deliver his lecture. Now, I would like to conclude the webinar, and in last, not at the least, I would like to invite Dr. Johnny Kumar for vote of thanks. Dr. Johnny Kumar.
Thank you, sir. I am audible?
Yeah, audible.
Myself, Dr. Johnny Kumar, and good afternoon to all. On behalf of organizing team and our colleague CCR PG, Colin Yapanagar, I take this opportunity to propose the vote of thanks for those who have directly and indirectly contributed to this webinar. At the outset, I thank our chief guest and keynote speaker, Professor Mal. Sir, we are really enlightened with your knowledge and presence. And we are highly thankful to our keynote speaker, Dr. Muhammad Akram sir, Dr. R.K. Mishra sir, and Dr. Shankar Ganesh sir. Sir, your willingness to share your time and expertise in area of biological control and biopesticide was critical to the success of this webinar. And I also would like to thank our principal, Professor Naresh Kumar, for his support and motivation. Without him, this program was not possible. Special thanks to the organizing committee, Engineer Sudhir Kumar, teaching, non-teaching staff for their support and motivation. Our heartfelt thanks to our students for their active participation. With this warm word and a kind message, we move to the end of this webinar. Thank you very much. Thanks to all of you.